Okay, so to try to be a little creative and switch it up, we decided to make ours Italian themed, as you can see. And so this is our DNA molecule, but obviously we could not represent the double helix that DNA is supposed to be in the form in because of our food restrictions. But right here, represented by the pasta, is the double helix form that the DNA should be in. But we have it in the in a line. So now there are individual nucleotides right here, which are made up of a nitrogenous base, a 5-carbon sugar, and a phosphate group. The bread represents the 5-carbon sugar known as deoxyribose. The mozzarella balls right in between represent the phosphate groups, and the peppers and sausage represent the nitrogenous bases. The red pepper represents adenine, the green pepper represents thymine, the yellow peppers represent guanine, and the sausage represents cytosine. Now, between the individual nucleotides, there are covalent bonds, and connecting the nitrogenous bases are hydrogen bonds.
Between cytosine and guanine, there are 3 hydrogen bonds, and between adenine and thymine, there are only 2 hydrogen bonds. And also, each individual step of the ladder, the nucleotides are of equal width, because in each one there is one purine, which is a double ringed structure, and the purines are guanine and adenine. And there's also a pyrimidine, in each, and that's only a single ring structure. And the pyrimidines are thymine and cytosine. And there are also base pairing rules that the DNA molecule follows. For example, adenine will always pair with thymine, and guanine will always pair with cytosine. And this is important because when the DNA molecule replicates, each strand acts as a template for a new strand to come in and connect, because one strand will always have a complementary base sequence to the other strand.
Alright, so now I'm going to talk a little bit about DNA replication. So to start off, let's begin with the DNA molecule. We have one DNA molecule here, and it will be split up by a heliocase to begin replication. This will come up breaking the hydrogen bonds between each strand, like so, and will form something called a replication fork. The replication fork is the Y-shaped section between the two now separate strands of DNA.
So now, what happens here is that now we have our DNA molecule that's split up into 2 separate strands. Now one of these strands is considered a leading strand, and the other one is considered a lagging strand. This is because an enzyme called DNA polymerase goes up the leading strand, and we know it's the leading strand because it follows the way the helicase went, because it goes in an order of carbon prime 5 to carbon prime 3 on the deoxyribose, while the other, and it would go all the way up here following it. However, the other one, the laggard strand, the DNA polymerase, since the helicase is only so far up, it has to start at different sections to do what DNA polymerase does, which is add nucleotides that are found inside the nucleus and pair them up and pair up the nitrogenous bases with its complement. And so now what happens here is that since it can only form part of it because it has to start at a separate origin, the fragment that will form here is called an Okazaki fragment. And now
these Okazaki fragments only appear on the laggard strand.
Yeah,
right here.
These are only on the laggard strand, and there will be multiple Okazaki fragments throughout the laggard strand as helicase goes further up, and DNA polymerase has to start at multiple different origins. And so what will happen at the end of this is that an enzyme called DNA ligase will come and fit the Okazaki fragments together, forming a strand of DNA.
Now, just to show you guys how this is going to work, because this is the laggard strand, this forms Okazaki fragments represented by these 2 nucleotides. So these 2 will bond first. Then let's say These three are another Okazaki fragment. These three will bond next. Now, because this is the leading strand, they go on one at a time, following the DNA polymerase. There we go. There we go. Oh. This process will repeat until there are 2 separate strands of DNA.
And this is
called semi-conservative replication because out of the original 2 strands, 2 new strands are produced. So half the strands are conserved and half the strands are no.
And these 2 represent the old strands of these 2 represent.
You can see with the different colors of red.
Thank you guys.
Thank you guys.
Thank you.